Welcome, everyone. I'm Rob Sweet from the Department of Urology and Executive Director for the WWAMI Institute for Simulation in Healthcare here at UW, and CREST, our Center for Research and Education in Simulation Technologies. And it gives me great pleasure and honor to be able to introduce to you a good friend, a colleague, and a true pioneer, and that's Dr. Michael McAlpine from University of Minnesota. He's an associate professor there. He's a named professor. He is actually a 2017 Presidential Early Career Award winner for scientists and engineers. And if you look at his CV, he really is at the top of his field. And I'm excited to bring him here really to introduce and to explore and discuss the role that 3D printing has in medicine and healthcare and what they're doing right now as applications for it, and how he's really part of the group that's pushing the field. And when we talk about simulation of devices or of human tissues, especially in our world in simulating for training, the biggest issue we come across is fidelity. That is a key issue that we struggle with. And 3D printing really promises to not only solve some of the problems of cost, but also of improving anatomic and tissue fidelity. So, and also providing functionality in physical materials, which is really exciting. Obviously, the body is a dynamic model, and we need to replicate it in the same way. So with that, I'd like to bring Dr. McAlpine up, and we're really excited to hear what you have to say.
Okay, well, thanks everyone. You can hear me okay? It's good. Thanks for hosting me here today. Lots of people here during the summer. Thanks for meeting with me. As Rob mentioned, our program at the University of Minnesota is based around trying to extend the capabilities of 3D printing beyond the hard plastics that you would traditionally print with a commercial 3D printer to functional materials. And for us, functional materials means electronic materials. It can mean cells, biological materials, biomaterials, the integration of these materials together. So on a single platform system, they can print both electronics and cells, and maybe even hard plastics too, and other types of capabilities, which I'll demonstrate today. And as of now, this is not something that's commercially available. This is a capability that we built in our group. We are in the mechanical engineering department, so I do have talented students who know how to build things and do the programming. And they can basically build these unique printers, which for now we have a mini monopoly on this, but who knows how long this will last as people continue to enter this exciting field. But I think there's a lot of possibility, a lot of potential, a lot of things that could be done here. And what I'm really going to demonstrate is kind of our overarching vision for this field, but then also some of the actual projects that we've accomplished so far in it. And they're very diverse projects. In some sense, they're almost completely indistinguishable one from the next, but they're all tied together from the fact that it comes together with this 3D printing tool, technology that we built. Now, I should mention that even though I'm in the mechanical engineering department, I am a chemist by training. I received my degree in chemistry. I'm sort of a material scientist in a sense, and so we're we know a little bit about many different things, which allows us to collaborate with folks and have this 3D printing core as our foundation. I think one of the things that I've always been interested in, if we think kind of very far out, is some of these technologies companies like Boston Dynamics are basically building these robots that can behave more and more like humans. And then the question is, this is sort of a mechanical engineering problem, a computer science problem. But what if you could kind of approach this problem from an inverted perspective and take humans and make them more like robots and start to integrate electronics and functionality onto the human that can allow capabilities that extend beyond what humans ordinarily can possess. So this can affect some real world problems in biomedicine and bioengineering, such as smart prosthetics, regenerative applications. But you can also sort of think outside the box too, in terms of augmented functionality too, and I will talk about that today. And sort of the classic example of a electronic device that's actually interwoven on the body would be something like a cochlear implant, where someone loses their hearing completely, and then they have this wire that's thread through the cochlea that allows them to restore the hearing too by basically having this wire stimulate the nerve endings within the cochlea, do a Fourier transform, and then send those signals to the brain that allow them to hear again. But it is an invasive procedure. There is a battery mounted on the skull. There's complications associated with potential infection with the threading the electrode through the cochlea. So if we can make this interface more seamless, a more seamless interface between the electronics and the biology, this could allow for many different types of applications. And even now, many people in the audience probably wear these smartwatches, which are basically electronics tethered to their wrist. If you can make that a more seamless interface, that can open up a whole range of possibilities, some of which I'll discuss as we go forward. So, in some sense, this is sort of an electronics-focused goal, because I do think electronics builds a lot of functionality that we're used to. But then the question is, how do you take electronics? And you look at electronics in the traditional sense, and you look at biology. What are some of the distinctions between these two that make it difficult from a materials perspective, a basic materials perspective, to merge electronics onto the biology in a seamless format? And I can think of sort of three different distinctions, three dichotomies here that make this a challenging goal. The first thing is that the electronics, the wafer, the silicon wafer, even the smartwatch, your laptop, your cell phone, they're all basically a two-dimensional flat format. All the processing that Intel does is based around two-dimensional fabrication and processing. Whereas biology, from the DNA level, the protein level, the organ level, the organism level, it's all three-dimensional. So you have this dimensionality mismatch between the electronics and the biology, which make it difficult to merge them together in a seamless way. Second thing is there's mechanical mismatch. The electronic wafers, they're based on silicon, single crystal silicon. They have a Young's modulus around 100 gigapascal. They're hard, rigid, brittle. If you drop them, they shatter. Biology is quite different. A lot of biology can be soft, flexible, not just flexible, but also stretchable, which is another mechanical mode that's quite distinct from the brittle wafers. And so you have this mechanical mismatch between the two that makes it difficult to merge these in a seamless way as well. The third thing is that the silicon requires high temperatures to process, about 1,000 degrees C, which is obviously well beyond what biology can withstand before it degrades or decomposes. And so you have these thermal restrictions between the high-quality single crystal silicon that gives you high-performance computing and the biology that, again, make it difficult to merge functional materials onto the biology. Now, people have come up with a lot of creative approaches to overcome these dichotomies. For example, John Rogers at Northwestern has done a lot of work on taking two-dimensional fabricated wafers, peeling off the electronics from the wafer onto a platform that's flexible, maybe even stretchable, by having these kind of accordion-shaped silicon ribbons, and then merging it onto the biology as a final step to actually implement electronics directly on your wrist. And the thing about that is that's led to a lot of amazing advances, pioneering advances. And the one thing I should mention, though, is that interface there is a two-dimensional, sort of pseudo-two-dimensional interface, whereas, again, it'd be nice if you could actually interweave electronics with the biology in 3 dimensions. And it would also be nice if you could decouple from the electronics microfabrication processing and have something that you could carry around in your backpack with you to make electronics on the spot. Let's say you're a soldier out in the field and you just want to print an electronic circuit. You don't want to wait for Intel to fabricate one and then airdrop it to you. So that's where I think the 3D printing comes into play. It gives you an extra dimensionality. It gives you autonomy from the conventional microfabrication processing. And the only thing about this, though, is that a commercial 3D printer, the ones that you go out and purchase now, they're basically, you feed in a filament, a polymer filament, it melts that filament, it extrudes it, and then it solidifies and cools down to a hard plastic object. And now you can buy advanced 3D printers that could do both red and blue plastic and merge them together. But what we'd like to do, I think, that makes 3D printing a revolutionary process is to go beyond the hard plastic, hard plastic has many utilities, but go beyond the hard plastic to electronics, to cells, to the merger of these materials together in 3 dimensions. And this requires that you build 3D printers from scratch. And so the one that you see in the background here is actually our 2nd generation printer that we built. And you can see this is a 4-nozzle printer. We have imaging capabilities. We have our pressure dispensers over here. Everything is computer controlled, including the stage. And this printer allowed us to do many different things, but actually now we're on our third generation, which I'll show in a bit. But this does actually address all three of these challenges that we see here, because we can print in 3 dimensions. So we can print in a dimensionality that matches the biology, is commensurate with the biology, but also can be patient specific and match the biology with high fidelity, as Rob mentioned. The second thing is we can print functional materials, functional inks, but then merge them also. They can be inorganic inks even, but then also merge those with soft inks. And these soft inks can be even orders of magnitude softer than the hard plastics in a commercial printer. And so now you can overcome some of these mechanical discrepancies as well between the electronics and the functional materials and the biology. And the last thing is that we don't use heating when we print. Actually, we don't do this kind of heat extrusion melt. We extrude at room temperature most of the time. And so a lot of our extrusion is done at room temperature, so we can process materials at high temperature. We can then formulate inks from them. But then the extrusion process and the extrusion of these inks is done at room temperature, or maybe slightly elevated if you want to include cells there and make them happy by using heating jacket. But we can basically do much lower than the 1,000 degrees C that you would need for a silicon wafer. And so you can overcome a lot of these thermal mismatches that allow you to merge the electronics with the biology. So that's kind of that's my vision for 3D printing. This is a unique vision. I haven't heard many people talk about this, but I do think this is the future, at least in my head. And I think we've made some progress on a number of different projects which show this. And I think that the validation of this so far is the fact that, you know, we've at the University of Minnesota, we set up these printers when we arrived 2 years ago, and then people just came out of the woodwork wanting to collaborate with us on this, because this is not something that you can do with a commercial printer. And so, you know, suddenly we have one project on spinal cord repair, one project on printing LEDs arrays, one project on soft robotics, many different types of things. It keeps me busy because I have to learn about every single field that's out there. But the good thing is that this allows us to forge new collaborative relationships, but then still have at our core this unique capability that we're building every single day as we pursue these projects. So one thing I should mention here also is about the manufacturing. So, you know, one downside of 3D printing is the fact that it is a serial process, so it can be a bit slow. But there are some manufacturing advantages to the process as well, especially when you think of it from this perspective of printing functional materials. And I think the key advantage is really the multifunctionality, but then also the multi-scale approach, because the inks that we formulate, the functional inks, they typically are derived from some sort of functional nanoscale material, but we're not actually printing at the nanometer scale, because it's difficult to extrude something, force something through a nanometer scale nozzle without that nozzle clogging or breaking. So we don't try to print nanometer scale lines. Most of the printing that we do is on the, at minimum 10 micron, but typically 50 micron scale and above. That's our line width. Of course, the precision of the stage can be sub 100 nanometer in terms of the XYZ resolution of the stage. But what actually comes out of the nozzle and extrudes into a line is typically on the 50 micron scale and above. But that's okay, because every biological problem that we've worked on so far with this technology, that length scale has been sufficiently small that is commensurate with the biology. Although if we do want to go small in the future, we will have to think of some clever strategies around that. And then the last thing, of course, is that you build up these devices that you can take photographs of. You can hold it in your hand. These are macroscale objects. These are true manufactured objects. So you do have this multi-scale manufacturing from the nano ink to the microscale printing to the macroscale device, and it is multifunctional as well. So that's the manufacturing advantage of this approach. In addition to the fact that it can be made patient-specific with 3D scanning, and it can also be made autonomous, and then maybe you could put these things in your backpack, take them with you with only raw materials, basically print any type of device that you want. So what you lose in terms of mass production, you gain in terms of all these other types of manufacturing advantages. So just briefly, I want to talk about where this all got started. This was kind of a fun project we did about 4 or 5 years ago. I hadn't heard of 3D printing at all until 2011. I saw Hod Lipson. Of course, it's been around since the 80s. Chuck Hull invented it. But I hadn't heard of it until 2011 when I saw someone give a talk where they weren't printing hard plastic. They were actually printing flour and food coloring. They were printing food. And so he printed a muffin, and then you cut the muffin in half, and there was a letter written on the inside of the muffin. So it was an espionage muffin. And so I saw this talk, and I was like, this is an amazing tool. Maybe you can went back to my student, think of something where you can merge lots of different functional materials together using this tool. And he came back to me, and he disappeared for 2 months, and then eventually I called him to my office and said, "Where'd you go?" And he said, "Well, I came up with this idea, but I thought it was a stupid idea, so I was trying to think of something else." And then he told me what it was, and I thought it was an interesting idea, which is, can you print an organ, but then sort of as the organ grows, can you also print electronics within the organ as well, which is three-dimensionally interwoven throughout that organ. Now, people have been using 3D printing at this point for tissue engineering, but for example, with ear and with cartilage tissue. But what they were doing is they were 3D printing a hard plastic mold, and then within that mold, they would then put their cell-seeded hydrogel and do the tissue engineering. What we wanted to do is print the cells directly from the printer and then also print the electronics as well in a three-dimensional format where a mold is not determining the shape of the final design. It's actually the computer that's determining the final shape of the object. And, this is all very simple. This is very crude electronics. This is crude tissue engineering. But it did kind of get us off the ground and led to some of the more sophisticated projects, which I'll show later. And for this particular project, this is our first generation printer that we purchased from this company, Fab at Home. They don't exist anymore. It was a simple $2,000 plastic printer. A high school student helped us set it up. We couldn't go and buy one of these expensive printers because you can only put proprietary polymers into those printers. And if you put weird stuff into it, like silver particles or cells, it'll jam pretty quickly and you violated your warranty because you put stuff in there you weren't supposed to. So buying one of these $2,000 printers in the lab for this kind of fun project, I think it was kind of a good start. I didn't know it would lead to bigger things. It was just something that we thought would be fun as a one-off project. But and of course here what we're printing chondrocyte cells within an alginate matrix. Again, very basic tissue engineering. It's not something that, from a tissue engineering perspective, it's not something that breaks new ground in tissue engineering or cell biology per se. But it's really just this demonstration that you can extrude cells from one of these printers, and you can also print electronics as well. The cell death is typically from the shear forces associated with forcing these cells through the nozzle. But with robust cells like fibroblasts, chondrocytes, you can get 90% viability. And now actually we're even printing more delicate cells like neuroprogenitor cells, and we are also getting pretty high viabilities with those as well, although it took a lot of effort to get to that point. But the concept here is you have this hydrogel that you've printed, you have these chondrocytes, you print these with the electronics interwoven, and then you culture it after the printing process into some sort of tissue or neocartilage type of tissue. And then what you can see here is that you can see this coil in the ear. This is electrically conducting inductive coil. And it looks like just a flat coil, so you're thinking, where's the three-dimensionality to it? But in fact, there are cells on top of the coil, and also the coil penetrates vertically down through the tissue and then comes out through this spiral-shaped electrode on the side. So you do have three-dimensionality. You have horizontal electronics, vertical electronics, spiral-shaped electronics, with the electronics sort of merging with the biology in 3 dimensions. Again, a very crude concept, but I think an interesting first demonstration of what's possible here. And you're probably thinking, well, this doesn't look anything, it's not quite acting like a real ear, doesn't quite look like a real ear. But actually, we came up with this kind of fun concept, which is, you think of your cell phone now. Your cell phone is an electrical device, electronic gadget. But in order to communicate with your brain, which is also electronic, none of your 5 senses are direct electronic to the brain. So what your cell phone has to do is convert the electronic to acoustic, the speaker, mechanical, and then your ear converts the mechanical back to electrical to go to the brain, because you have little hair cells that vibrate in your cochlea. They act as a little Fourier transform. So it'd be great if you could take your cell phone and then just plug it directly into your brain and have basically direct electronic communication. And none of your 5 senses do that. We have smell and taste, which are chemical interactions that go to electronic. We have touch and hearing, which are mechanical. And then we have sight, which is photonic to electronic. And that's because when we evolved, we weren't surrounded by electronic gadgets like we are now, where when I teach my class, everyone has their laptop in front of them. So if you could just go straight to the brain now with a 6th electronic sense, that'd be useful. And in fact, people who do have cochlear implants, they can take their cell phone and plug it into the implant and get direct from phone to brain type of connection. And actually, Rob invited me to a Society for Laparoscopic Surgeon talk back in December, and I didn't tell Rob this story, but after the talk, the head of the Society for Laparoscopic Surgeons came up to me and said that he actually wears a cochlear implant, and he said he's never taken his cell phone and plugged it directly into the implant. And so I said, well, I'm sorry if I offended you, but he said there's no need no need to do that because I have Bluetooth built into my cochlear implant, so I can just Bluetooth from my phone directly to my brain, basically. So basically, this concept is not science fiction. It's really possible. In fact, this ear can also, because in inductive coupling, it can go beyond the normal range of hearing, which is 20 kilohertz, maybe up to the megahertz or so. And in fact, you can see here, these are two, this is a left and right bionic ear that we 3D printed that are sitting here and listening to Beethoven in their spare time. So this was kind of a first project, one-off project, but we've gotten a lot more sophisticated since then, which I'll show. Actually, one of the projects we're working on now with Rob, which we're set to publish, does have to do with kind of the low-hanging fruit for 3D printing, which are creating some of these organ models. But there's some drawbacks to the current 3D printed organ models. Again, they're hard plastic objects. A surgeon, if he wants to practice on them, they're difficult to cut into. And if you look at the statistics, there are several thousand of these surgical never event claims that occur every year. Those are surgical events that are not supposed to happen. Like just 2 days ago, you probably saw the article about man had his testicle removed, then they removed the wrong testicle. And these things happen quite frequently. Actually, this British Medical Journal article, it's a little bit controversial, but it claims that medical errors are the third leading cause of death in the United States. Now, whether that's true or not, I'm not sure, but one of the problems here is that the doctors have to rely on CAT scans and MRI scans, which are basically two-dimensional scans, and then piece together in their mind the three-dimensional structure of the object before the surgery. They have to do a lot of improvisation on the spot. So it would be nice if you could create a 3D model which exactly represents the exact feel, touch, tactile sensation of the organ, and maybe even make it like a game of operation where as you operate on this organ model, do the surgical practice, it will respond electrically to your surgical tools. So some sort of advanced organ model. That's the idea. And if you look at some of the 3D printing inks that are out there, some of the filaments, the Young's moduli of these filaments, they're typically, the soft ones are typically on the order of the megapascal range. So even NinjaFlex has a shore hardness of 40, it's basically about 15.2 megapascal in terms of Young's modulus. This is well beyond orders of magnitude higher than the feel and touch of organ and tissue, the Young's modulus of actual tissue samples. So we thought, actually it was really Rob's idea. Can you create an organ model which has the exact feel, the exact Young's modulus, patient-specific in a sense, of the original organ to create a tool for surgical practice which mimics as much as possible the original organ. In this case, we started with a simple organ, which is a prostate sample. And then later on, we came up with this idea, can you embed it with 3D printed sensors that can then respond to touch in the ways that I described before? And it turns out that this actually worked out pretty well. So first of all, we did find a patient. We got MRI scan from the patient in order to figure out the anatomical structure of these models. We then 3D printed these models using our custom 3D printing platform. So these are inks that we developed in our lab, which in principle should have much lower Young's moduli than the commercial inks. And so this is a soft ink printed in the form of this prostate here. And there's also a sacrificial ink there too, that's the white that we cut away later on. Now what we can do is we can take this organ model that we've printed and then do an MRI scan on the organ model itself. So this is actually an MRI scan on the organ model. And then that way we have a digital representation of both the original organ and of the organ model. And we did do some software calculations, some comparison tools to figure out exactly the anatomical fidelity between the organ model and the original organ. And these are surface calculations. They're not full point calculations, but they're surface calculations. But the overall anatomic fidelity using these 3D registration techniques was found to be about 98%, which is pretty accurate. Now, in addition to that, we wanted to do mechanical tests, get the exact mechanical Young's modulus of these inks, develop these inks so that they can be patient-specific, In order to do that, we did have to go and take the tissue samples themselves and do some mechanical, both static and dynamic mechanical tests on the tissue samples from the patient in order to determine their properties and then try to match them with our inks. And we also did hardness tests and optical tests. And one of the nice things about being at the University of Minnesota is the fact that the medical school is across the street. So, I asked my student, I said, you need to go get a bunch of human prostate samples for this experiment. Then 2 days later, he got a bunch of human prostate. I'm not sure exactly where he got them from, but no, we followed all IRB protocols. But we did these tests on them, and then what we can do is we can compare these compression tests and these mechanical tests on the tissue samples to the organ model itself. And these are not just physical tests. We also did actually a fair bit of modeling for this paper as well. But what you can see here is actually a pretty close representation and fidelity, again, between the mechanical tests of the inks and of the tissue samples that we obtained. We obtained 3 prostate samples and 3 different inks that we developed. And you can see in the low strain region, at least, you can see almost a perfect match between the 3 inks that we developed here and the 3 tissue samples that we obtained. So the Young's modulus here is about 20 kilopascal on average. So again, orders of magnitude less than the Ninja Flex that you would find with a commercial 3D printer. It's kind of strange because you hold these things in your hand, and it really does feel like you're holding a real organ. It's so soft and has the exact tactile feel of the organ itself. We can also add dyes to it, of course, that can do color matching as well, which is one of the things we did. It's pretty much a color match to blood. Now, we did also do surgical practice on these organ models. So you can do things like put endoscopes into them. They have a 3-dimensional structure. They have both interior structure as well as exterior structure, which is anatomically matched. So you can see here the prostate urethra here. And, So what we did here is we had another student in my group who was working on 3D printing tactile sensors and touch sensors. These are capacitive, but we've also done piezoresistive. So you can integrate these 3D printed sensors on the model as well. So as you do the surgery, for example, it can respond electrically to the touch of the grasper, the touch of the scissors, the touch of the endoscope, and give you an actual quantitative feedback to the surgical practice that you wouldn't get from, again, from a hard plastic object or from an MRI or CAT scan. So again, it's like sort of like a game of operation. And this can be both on the surface of the organ model, or it can be integrated into the interior of the model as well, into the urethra. So kind of this combination of different materials that are not capable at all with 3D printing, soft, ultra soft polymers, functional polymers, electronically active devices, all being integrated together in this kind of advanced surgical practice tool that we've developed here with Rob. Okay, next thing I would like to discuss here is sort of our bread and butter. This is sort of where we're really pushing the limits of what's possible with 3D printing, which is 3D printed electronics. This means both conducting electronics and semiconducting electronics. So to give you an idea of the complexity of what we can accomplish now with our advanced 3rd generation 3D printing tool, this is a paper that came out of Advanced Materials just about a month ago, and really took off. Actually, I was pretty excited about it, the response to it, where we're now creating multiple layers here of multifunctional inks and materials being integrated together into these kind of complex device formats. In this case, this is a touch tactile sensor, which is piezoresistive. So we have a bottom electrode, a top electrode, and a coil that's printed here, which is touch sensitive. It's a formulation of nanoparticle inks within an elastomer matrix. It responds compressively to touch with a resistance change. And you can even do things like create sacrificial supporting layers here between the electrodes that can then be washed away in the end. So you have a freestanding eye-shaped geometry device here in the end. So very complex. We're now getting pretty sophisticated in terms of the types of printing we can do. And I think nothing says this more than the video I'm going to show here, which is a multi-head printer that we developed printing directly on this model hand. So this is not a real hand, of course. This is a model hand. My students right now, they're too scared to put their hand, but I'm going to force them at some point. And so this is, you can see the multi-head approach here as it rotates between multifunctional inks. Some of these inks are conducting, some of them are silicones. There's the piezoresistive coil in between. And this is the sacrificial supporting layer. So one thing you're not seeing here is the fact that this finger that you see is actually a curved surface. And what we did beforehand is we 3D scanned that curved surface, and this device is actually being conformally printed. It's hard to see here, but it's actually adapting to the curvature of that finger as it's conformally printed onto the surface. And in fact, if you want to print on your real hand, you have to take into account the fact that your hand is going to vibrate a little bit. So now my students actually I was one talented student who's developed an algorithm that can track motion of a hand in real time in order to do this. And he's even shown me amazing videos where he's taken paper and he's crumpled the paper, and then he uncrumpled it, and then he can track the topology of the paper by doing a scan and print type of approach. So it's getting kind of fancy at this point. Now these are conducting inks that are basically fabrics. So this is a particle, silver particle, within a elastomeric matrix. You can see it can be stretched up to 3 times. So a stretchable fabric, where the mechanical properties of this fabric depend on the silver content loading within the elastomeric matrix. But the key thing here is that you can pick a silver composition for that middle coil, which is exactly at the percolation limit of the particles within the elastomeric matrix. So that gives you the most sensitivity to touch and to tactile sensation. If you pick this 68% loading here, you can see they have the largest change in resistance. And we can actually get gauge factors of these devices up to about 180, which is almost on par with a single crystal semiconductor. So you can see here that this device is sensitive enough that you can take it and you can merge it onto your wrist, as you can see here. And my student was able to measure his pulse in a sedentary state. Then he went running around the block and came back, and you can see that the amplitude and frequency increases with the increased pulse frequency here. Now, one other thing we can do is we can integrate these into device arrays. So this is not just a single tactile sensor. This is an array of 5x5 tactile sensors here, which was entirely 3D printed. And then you can take this glass triangle here, push it into that device array, and map out the shape of the triangle into this array of tactile sensors that's being developed here. So we're not where Intel is really, but we are actually getting pretty sophisticated in terms of device integration, device capabilities, material capabilities. You can see in real time here, this is similar to what we saw with the organ model. This is a tactile sensor on this hand, where every time he touches it, you do get this electrical feedback response from this touch sensor that was directly 3D printed on this model hand. And I think the way this is going in the future is you're a soldier out in the field. You need a solar panel to charge your weapon. So you take your 3D printer out of your backpack and you print a solar panel directly on your wrist, or you print a chemical sensor directly on your wrist that can detect chemical warfare, something along those lines. So direct 3D printing of devices on the body. I think that's a killer application that really not many people are talking about, but I think is going to be a big thing going forward. People at home are going to print electronics directly on their wrist. They're going to do home surgery with their home 3D printer. Lots of exciting things. Now, we are actually the first group to not just print conductors, but we've also 3D printed semiconductors, semiconducting materials, semiconducting devices. Conductors can only get you so far, but a semiconductor is where you can do things like processing, light-emitting diodes, complex circuitry. And so 3 years ago, we had this idea of 3D printing an LED, which is a light-emitting diode, a semiconducting-based device. And we were able to do it. This was the first demonstration of that fully 3D printed LED. And then Jennifer Lewis is one of the pioneers in the 3D printing field. She was nice enough to write this little Nature article about it about 6 months after the paper came out. Now, the trick here is that, we go back to this whole thing about 3D printing. What can the current commercial tools do? They can print hard plastic and blue plastic, merge them together. But to print an LED, and this is one demonstration here, there's many different types of materials that need to come together. So we have a bottom silver nanoparticle electrode. We have whole transport and injection polymer layers. We have a inorganic semiconductor layer. These are inorganic CAD selenide nanoparticles, and a liquid metal top contact on top to act as your top contact. So you're talking about liquid metals, solid metals, hydrophobic materials, hydrophilic materials, organics, inorganics, basically all coming from the same 3D printer, which occupies a space that's about the size of this podium here. So, this is again, we go back to Intel. Intel can do one material with basically silicon. This many different types of materials being integrated together in this desktop tool. So I think there's a lot of advantages here. This doesn't even include the biological, but there's some tricks to doing this. So I should mention that to 3D print a semiconducting ink, you have to be very careful about your ink formulation, because if you just deposit a droplet of particles on a substrate, it'll dry and all the particles will spit out into this coffee ring. So what we do is we use tricks from chemistry using low surface tension co-solvents. So as the droplet dries, it actually creates a uniform distribution of these particles throughout the droplet. And that gives us uniform device properties. And actually, one of the companies that's sponsoring us now is Boeing. And they do want us to create a large-scale array of 3D printed LEDs on a flexible platform. And so we're actually able to do that now by controlling this uniformity of this process here. So as you can see here, this is a fully 3D printed green LED, a fully 3D printed orange LED. You get nice color purity, good color saturation. Now the key question is, what is the advantage of 3D printing semiconducting-based devices over using a traditional microfab tool? What is the purpose of doing this? And I think that the key advantage here, again, is the merger of high-performance electronics with biomedical devices. That's really where the advantage is. Because Google, for a while, has been interested in how do you take electronics and merge it onto a contact lens. You can take metallized electronics and create an antenna on the outer surface of the contact lens. That's easy to do. But to create semiconducting electronics for visualization, for other types of technologies on the contact lens is much harder to do. It's difficult to take a semiconductor from its rigid planar format, merge it onto a three-dimensional soft biomedical device like a contact lens. But again, because our 3D printing process is additive, we can deposit the device layer by layer on the surface of the biomedical device or within the biomedical device in a way that can't be done with any type of traditional microfab. And the trick here is we also, again, we go and we 3D scan the contact lens. We get the exact geometry of the lens, and then we conformally print these semiconducting-based devices onto these onto the contact lens. Here is a fully 3D printed functioning orange LED that was printed onto the surface of this contact lens here. And actually now my students are showing me things where now they can get 4 types of LEDs, and now we're doing photodiodes as well, integrated onto these devices. So huge potential for advanced biomedical devices that are not just polymers but also electronic functionality. You can build electronics into the third dimension. So a lot of the research we do is inspired by YouTube videos. This is one of them that I saw. We do a lot of work on cats and that sort of thing. But this is a movie I saw which fascinated with me, where someone built this at home in their basement, basically created this array, three-dimensional array of LEDs that could create this kind of three-dimensional geometry here. And I thought, can we 3D print a structure like this, where you can 3D print in 3 dimensions a multicolored array of LEDs, where the LEDs are 3D printed, the cube is 3D printed, everything comes out of the same 3D printer, taking electronics out of this planar rigid format of the laptop into some sort of soft 3-dimensional structure with many different types of materials all being integrated together. And in fact, my student, he worked really hard on this. It was an amazing achievement, but that's, that I think one of our really exciting breakthroughs here was being able to, it doesn't look as pretty as the image, but this is a silicone-based cube, which was 3D printed. And then within this cube, you can see green and orange LEDs that are 3D printed within this structure. And actually, if you know much about the 3D printing community, you know that it's not just that printing the LEDs that's a big accomplishment here. Actually, the 3D printing people are now trying to figure out how do you print silicone. So, they're trying to print out how do you print soft polymers like silicone. Here, silicone is just the matrix in this cube. And then within that matrix, we actually have fully 3D printed semiconducting electronics built within this soft matrix. Again, all from this simple tool that costs about $15,000. So you can see here there's liquid metal droplets, the silicone base, the different colored LEDs, these electrical leads that come out the side. Many different materials being integrated together. And so I think this integration of materials, going beyond hard plastic, this is really going to revolutionize many different aspects in 3D printing as we go forward. Okay. Now, we do have some limitations on time, but there's just two more things I want to talk about that I'm pretty excited about. The bioengineers actually, they love this project, which is, I think one of the underrepresented tools in your tool set of what 3D printing can do is not just biology, not just electronics, but I think also chemical and biochemical control is something that can give huge advantages in terms of dynamic tissue engineering, dynamic bioengineering processes. And this is something that's difficult to accomplish, but something that we're starting to put our focus on. And we do have some collaborations in this area, actually, which is that we came up with this idea, what about printing drug delivery type of vehicles, things like vesicles or emulsions or whatnot. A lot of the times, the drawbacks of these emulsion-based techniques is that you do end up with a non-uniform distribution of particles or whatnot that have different volumes associated with them. You really want to get good uniformity, a good control over the dose of chemical you want to release, control over the time of when you release it, and control over the spatial orientation of where those chemicals are within the bioengineered matrix. And this project actually had nothing to do with 3D printing when we started, but then eventually we realized that this 3D printing gives you these advantages because the droplet that comes out of the 3D printer is well defined in terms of volume. So you have total control over the dose in your diffusion equation. You can then 3D print a shell on these things to encapsulate them, and you have control over space because the three-dimensional control of the 3D printer. And then my student came up with this really clever idea of within the shell of these particles, he puts these gold nanorods which vary by shape. And so the shape variation gives you a photothermal resonance called the plasmon effect here, where they can be triggered by specific wavelengths of laser light to then rupture the shell and then release the contents of the core through that hole that's been generated. So one crazy way to think about this is, let's say you have a collection of stem cells within a matrix, and you shine a red laser on it, and now it differentiates into a heart, and you shine a green laser on it, it differentiates into a liver, something like that. Obviously, we're nowhere near that, not even close, but although we have shown now differentiation using these capsules and cell migration as well using these capsules, these are ongoing projects. But the trick here is that this should be orthogonal, so the red laser should only trigger these guys and not these ones. The green laser should only trigger these guys and not the other ones. So you have selective response, you have control over dose, you have control over space, you have control over time of release as well. So really a beautiful bioengineering system for control to sort of mimic the orchestrated developmental events that go on within the body during developmental processes. And so again, we can 3D print these cores of some sort of biomolecule. Now, if you look at the volume of these cores, as promised, they're almost exactly half a nanoliter of volume from droplet to droplet, because the droplet that comes out of the printer is well defined. So you can see the tails of this histogram here don't even go to 0.4 or 0.6. It's actually a very precise control over dose that can be repeated from droplet to droplet in a multiplex way across a chip. And again, the multiplexing can be complex by virtue of the fact that we are using a 3D printer where everything is CAD designed here. So this is a complex drawing of a tiger here of two types of cores that are being printed in this multiplex manner. So what we do here is that we then encapsulate these cores with a PLGA shell, which retains the contents of the core. And we can even immerse, depending on what the core is, you can immerse these, in this case it's a dextran, but you can immerse these underwater for 24 hours and the contents of the core don't release within this limited time period. Although over a long time period you will get diffusion through the shell, but you can control the concentration of the shell to limit your diffusion. Now within the shell, we can introduce these gold nanorods. So our collaborator synthesized these rods for us. They have the same diameter, but they have two different lengths, a short rod and a long rod. And the short rod actually, they have two different resonance peaks here. So this is the peak that's determined by the diameter of the rods, and then they have two different lengths, so the peaks here are shifted one from the other between these two rods in terms of the absorbance spectrum. So the shorter rods here have a peak around 700 or so. The longer rods around 8 880. And you can see that where this one rod is a peak, the other rod is a valley. And so where this rod, the first rod is a peak, the other rod is a valley. So in principle, if you can pick your laser wavelength to hit right here at the maximum, it should only trigger heating of one type of rod and should not trigger at all the heating of the other type of rod. It should be an orthogonal system. Does that make sense? Same thing here. So the question is, does this actually work? And it turns out, surprisingly, that it does. So this is a capsule that we printed, a core shell capsule. You have your core, you have your PLG shell. Within that shell are these shorter rods. And then we trigger it with a laser that we also connect the laser onto a 3D printer so it can raster scan the rods into the matrix. And you can raster scan it over the capsule. What happens is the laser wavelength is tuned to the shape of the rod, so it heats up those rods. The rods melt the polymer shell, and then they form a hole in that shell, and then the contents from the core release through that hole. Okay, and what you can see here is if you raster scan it once or raster it twice, that changes the kinetics of release, but the actual thermodynamics of the release are diffusion controlled. Now, the trick here is that if you use a different wavelength laser that doesn't match the geometry of that rod, then amazingly you don't get any rupture whatsoever. So you really only have heating at that specific wavelength of the rod, and you go beyond that to a different laser wavelength and you get zero release through the capsule. And the opposite is true here. If you have the other size rods within the shell and use this other laser wavelength, then you can get trigger release, but then you use the first laser that worked on this case, and you get no release here whatsoever. So it's a truly orthogonal system. Two different lasers, one triggers one type of capsule, the other triggers the other capsule, and they don't crosstalk during this process. Now, this is true not just in 2 dimensions, but also 3 dimensions. So this is a 3-dimensional hydrogel structure that we printed, which we then embedded within these core shell capsules. And so there's blue ones and then red ones. And we can also do this triggered release in a 3-dimensional structure as well. And again, we're trying to do this now with, we've actually now 3D printed beating cardiomyocytes that were differentiated from a release capsules with Brenda Ogle's group at Minnesota. That's not published yet, but it's pretty exciting stuff that we're pursuing. Okay, now one last thing I would like to talk about here is this project where we start to bring everything together in a real-world biomedical project that hadn't been demonstrated before. And this is just one of several that we're working on. But peripheral nerve injury is something that a DARPA program manager said, can you use 3D printing for peripheral nerve repair? We hadn't seen, it turns out there were 1 or 2 papers, but we hadn't seen much on this at this point. And this was fascinating to us. It was a new area for me, and I learned about the peripheral nervous system. It's not that complicated. It's basically a 2-way communication system. So your brain sends signals out to the body that control motor function. And if I touch the podium here, that's sensory information that goes back to the brain. So this can be damaged in various types of ways. You can be in an accident. You can be a battlefield injury. You can have a degenerative disease like diabetes, which can degenerate the peripheral nerves. You can lose motor function. You can lose sensory, the sense of touch. Affects about 200,000 people annually. And we thought, can 3D printing be used to do peripheral nerve repair? And how exactly would that work? What would be a differentiating factor for 3D printing in peripheral nerve injuries? So we looked at a traumatic injury to the nerves. So for those of you who aren't familiar with this, as I wasn't a few years ago, but you think about a nerve here branching down your arm, and then you make a cut in that nerve, a traumatic injury to the nerve. You now have 2 ends of the nerve. You have the proximal end, which is still connected to the body, and then the distal end, which is kind of this free-floating end now. How do you bridge that gap and restore function and restore sensing and restore motor control with this nerve? So the way that this is done now is that the doctors use a technique called autograft, where you basically need a second surgery to take a healthy piece of nerve tissue somewhere else in the body and graft it into that gap to restore the function. And of course, this has many different complications because you have to sacrifice healthy nerve somewhere else. You need a second surgery that requires healing. So if you have a really nice friend who goes around donating nerves, then you can have an allograft which is basically a donated nerve sample that can then be grafted into that gap. But then there's other issues. You do need a donor. There may be some adaptation time required. So people have been working on these nerve guide conduits, which are synthetic analogs to this approach, which are basically polymer tubes. You suture the two ends of the nerve into that tube, and that acts as a physical guide to guide the regrowing nerve from the proximal end to the distal end. You basically have about 2 weeks to do that, because the nerve grows about 1 millimeter per day. The problem with these conduits is that most of the time they're not functional, they're just polymer tubes. And then second of all, the way that they make these tubes, they almost end up with these short, about 1 centimeter tubes that are linear. They can't really do much more complicated than that. So we thought, this actually opens a lot of potential here for 3D printing, because now we can maybe think about doing a patient-specific nerve guide conduit, which matches the geometry of the original nerve that we cut out. So we actually did do some animal studies here where we transected a branch point of a peripheral nerve in the sciatic nerve of a rat, where the branch point is actually a branching where you have a mixed motor and sensory, and it branches off of the motor on one side and then sensory on the other. We cut that out of the rat, so he loses both motor and sensory function. We wanted to restore both. And the trick here is we cut out the nerve tissue, we scan the nerve tissue, and then you can print a guide which matches the geometry of the original nerve tissue precisely, so you can restore the function there. So we did do this in rats. This is the sciatic nerve here, the branching point. We cut out this piece of nerve tissue. Here's the mixed side. As it branches down, it branches to motor on one side, sensory on the other. We took the nerve tissue. We did a 3D scan on the nerve tissue, get the geometry of that piece of nerve tissue, and then go and 3D print a conduit, which is not linear anymore. Now it can actually branch, just like the original nerve. Now this is a silicone-based conduit. This is not biodegradable. At least not at this stage, because the rats, you plant these things, the rats are actually running around for two or 3 months. So it needs to be robust enough, mechanically robust. But you can see we also can impregnate these things with proteins that can help direct the regrowing nerve as well and seal it off. So this is an opening of 500 microns and a length of about 1.2 centimeters. So the surgeon actually has to go and suture the soft nerve tissue within this half a millimeter opening. It's actually not that easy to do, but. Now, one thing I should mention here is that you can see the way we print this nerve guide. So we print it flat on a surface going this way. And you can actually think of two different ways that you can print the nerve guide. We can print it like that, the cylinder I showed before, where you're printing the structure like this. Or you can print it this way, where it's printing line by line going like you saw in the video here. So the question is, from a mechanical perspective, which is the better way to do it? And actually, the 3D printing, when people have been 3D printing ceramics, the same question came up, because if you print something layer by layer, does it have the same mechanical rigidity as a monolithic ceramic structure? The answer, of course, is no, because you're printing things layer by layer, cylinder, line by line, or circle by circle. So it doesn't have the same mechanical strength as if you print something line by line going this way, and you're stretching lines instead of cylinders. So in fact, we can do some mechanical modeling on this, figure out where the weak points of this guide are, add some reinforcement to it. We can go and look at these guides and understand we need to print them this way, laterally, line by line, because if we print them ring by ring, then what happens is they'll snap, and they'll snap at a lower yield stress here than printing it line by line. Okay, so there's some important mechanical considerations to the directionality in which you print these guides. Make sense? Yeah. Okay, good. Now, this is sort of, I think, you can think through this problem here, but then some things just happen in the lab that you weren't expecting. And one of those has to do with the fact that that there's not only a mechanical advantage to printing it line by line, there's also you can also build in cues into this device that you weren't expecting. So what happens in the body when you make this cut here is the distal end, that tissue starts to, there's a process called Wallerian degeneration. The tissue starts to get eaten away. And the Schwann cells that are left behind form these little tube networks. And actually, that's a good thing for the body, because if you ever do get regrowth of the nerve from the proximal end to the distal end, these actually act as physical guides to guide the regrowing nerve the direction it's supposed to go. It turns out that these guides have these same, our nerve guide conduits have the exact same type of structure within them, because we are printing it line by line. So our printing process is not perfect. So we get this artifact here. We have these little grooves built into our nerve guide conduit here. And these grooves can actually act as little grooves that can direct the regrowing nerve that is supposed to grow along the length of the guide and not every which direction in the guide. And this sounds like, is this really true? Is that really what's going on here? In fact, we did take neurons here and culture them onto a flat piece of silicone, and the axons grow in many different directions. But when you culture them onto this guide here, with these grooves built into the guide, you can see the axons do direct their growth preferentially along the grooves of that guide there. So a complete artifact of the 3D printing process actually allowed us to build in physical cues to help the regrowing nerve the direction it's supposed to regrow. That was an accident. This was on purpose. We can introduce protein factors, capsules like I showed before, into the guide that tell the regrowing nerve that it's supposed to branch at that branch point and that the motor is supposed to travel one way, the sensory is supposed to travel the other way. I don't think it's that simple. I don't think that's actually what's going on here, but that's kind of the concept of why we're introducing these capsules into the guide. And you can see that we have both red and green here, and then it branches green on one side, red on the other to direct motor one side and then sensory on the other side. And we did do some modeling on this. So the last thing I want to show, of course, is the actual animal study. So this is a preliminary animal study, but we did actually implant these things in rats. You can see without those chemical cues there, we do get some regeneration, particularly on the sensory side, but this is neurofilament staining. But we do get, with those chemical capsules that we imprint into the guide, we do actually get significantly more regeneration on both the motor and sensory side by having those capsules in there. For reasons we're not entirely clear yet, but with more studies we'll be clear. But this is, I think nothing says more than the video itself. So this is the rat one day after the surgery. So we cut out the nerve tissue from his back hind leg. You can see he's struggling to walk there. If you look at his back hind leg here carefully, he can move it a little bit, but not very well. And then 2 months after the surgery, after implanting this guide here, suddenly the rat can run and walk just as if he was brand new. Of course, the guide is still there. It's not biodegradable, but you do get this regeneration. I think one thing going forward that we're trying to do is help all rats walk again. Yeah, that's an important goal for us. But also, we're trying to go beyond the peripheral nerves now to also central nerve, which is much more difficult because now you have to supply not just the scaffold, but as mentioned, the neurons as well. So we have to 3D print neuroprogenitor cells within the scaffolds, and we're actually starting to make some progress on that, though it is a challenge. So, okay, so that's all I have to say. Many different types of folks help make this possible, collaborators, funding agencies. You can see the diversity of the projects here allows us to kind of tap into multiple sources, multiple different communities of people, and support. Always looking for more. And thanks again for having me here today, and I'm happy to answer any questions that you may have. Thanks a lot.
Thank you, Mike. That was fantastic. Very stimulated by that whole lecture. I guess one thing I'd like to first of all commend you for, and I think for the students in the room, I mean, this is a great example of someone coming from a chemistry background who's now been able to explore all these other fields, with mechanical engineering, bioengineering, biomedical engineering, neurology, neuroscience. And what I think you did really well is made it relevant to everyone in this room. I think you did an outstanding job in presenting that to make it understandable when you have a diverse group of people like this in this room to hear a talk like this. And so I really commend you on that. And students in the room, that's a great example of how to do it. I have one question, and then I want to open it to the floor as well. Is Cece here too? Cece, hi. So this is co-sponsored between bioengineering and the medical school, Wish and Crest. And so, Cece, I'm sure you have questions, or someone from your side of the house wants to. Yeah, sure. You're certainly welcome.
The question was, talk a little bit more about the stability of the printing directly onto the body, and then also, second part of the question was, can you 3D print biomedical devices such as the nerve guide directly into the body? And the answer is yes, that's where we're going. Now, when I say stability, what I'm referring to is actually the fact, let's say you had a real hand, your hand's going to be shaking, and so you need to track that in real time. But you do have to take into account the fact that the materials may be toxic as well, or you need to take account the stability of the materials, the toxicity of the materials. For example, the electronic device we printed on that model hand was based on silver particle ink. Now, it does cure at room temperature, so you don't have to heat it. You don't have to heat the hand, but the silver would be toxic to cells, to the body. We need to find a good replacement for that going forward. That's just a demonstration. But actually, I thought we were the only ones to do this so far with this direct printing. But then I got a little scared because just 2 days ago I saw someone give a talk at a conference where he had opened a rat and directly printed an early type of bone structure onto a rat. So people are starting to do this, push into this area. This is really the next big thing: direct printing of biomedical devices. I think this is important because, instead of taking let's say someone's in an accident, instead of taking that person as a patient to the hospital, if you can bring the 3D printer there, you can bring the hospital to the patient and 3D print the device directly on the patient by doing a direct scan and print on the spot using only raw materials. I think that's a critical area. So
you can picture some type of micro robot that actually goes into the area, is getting that information, and then actually laying down the materials.
That's exactly where I think this is going.